Hi, everybody. My name is Sandra Ribeiro. I am a communications advisor at the Canadian Light Source. And I'm very privileged to work with a group of very talented communicators from light source facilities around the world. I'm assuming because you're here, you know a little bit about lightsources.org. But this collaboration of communicators was established in 2004. We're going to be celebrating our 20th anniversary next year. So stay tuned for more events related to that. We have grown over the years. We now have 25 members representing 30 facilities around the world, in addition to the African Light Source Project. And really, lightsources.org is the website you can go to for jobs, news, events, proposal deadlines. So a great resource for everyone in the light source community. And that's it. I'm done. I'm just here to welcome you all and to introduce my very talented colleague, Cindy Lee, who is a communications specialist at the Advanced Light Source in Berkeley, California, and she's going to be our moderator for today's seminar. Cindy.
Thank you, Sandra, and thank you to lightsources.org for holding this special session. So now I'll bring on Julia and Steven. I also have a couple questions for the audience. I'll ask Stefania to launch the poll now. So this question is for you all who are joining us today. Do you consider yourself primarily a researcher, a communicator, both, or something else? So get your votes in. Now, I can't see how many people are voting, but I'm hoping Stefania will keep me posted.
Yes, I think we can end the poll and show the result. 100% of participants have now responded.
Fantastic. So it's looking like we have about 27% who are researchers, 45% who are communicators, 9% doing both, you're the real heroes, and 18% other. So, feel free to type in the chat what you do. And, especially the people who combine them or doing something else, feel free to share. I have one more question for you all. Where are you tuning in from today? So light sources, which is where I work, bioluminescence, which is what we're talking about today, that those can both be found in many parts of the world. So I think we also have audience members from all over the world. So I'd love to see where you all are tuning in from. And I think Julia and Steve might be curious as well. So feel free to put that in the chat. And while we're waiting for some answers, I would like to introduce the stars of today's event. So first we have Julia Kuo. She is the author and illustrator of Luminous, the book that we're discussing today, and the illustrator of several picture and specialty books, including the best-selling book, Rise. She's created editorial illustrations for publications such as the Wall Street Journal, the Economist, and the New York Times. Julia has taught illustration courses at Columbia College, Chicago, and at her alma mater, Washington University in St. Louis. She has been an artist in residence twice at the Banff Centre for the Arts and was a 2019 to 2021 fellow with the Gray Center for Arts and Inquiry at the University of Chicago. She currently lives in Seattle, Washington. We also have Steve Haddock, who has been participating in deep sea research for more than 30 years. He studies marine diversity, molecular biology, and bioluminescence at the Monterey Bay Aquarium Research Institute and UC Santa Cruz. He specializes in fragile gelatinous drifters that are abundant in the deep sea and open ocean. In addition to conducting research expeditions, he has documented many luminous species for the first time. He uses genetic methods to reveal the relationships between organisms and to understand the proteins and chemicals that they use to make bioluminescent light. He also runs the bioluminescence webpage, the Citizen Science Project, jellywatch.org, and teaches computing to scientists at practicalcomputing.org. So my colleague Silvana has put a bunch of links in the chat for you to check out more about our speakers. Welcome, Julia and Steve. So first, Julia has offered to share a passage from Luminous. So we'll kick off there.
Good morning, everyone. I'm so happy to be here with you all and to share a little bit about this book. I will read a little part of it. It has a main text and a subtext. So we'll get to, maybe a couple pages in. Luminous, living things that light up the night. When it's dark out, we need light to see. Light is a kind of energy that our eyes can see. Some light sources are natural, like the sun and fire. Other sources of light, like light bulbs, are created by people. But what if your body could make its own light? When a living creature, like a firefly, produces light, it is called bioluminescence. Bioluminescent creatures make the light through special chemical reactions inside their bodies. You could gleam on the forest floor. Foxfire or fairy fire is light made by bioluminescent fungi growing and glowing on rotting wood. Or shimmer inside a shadowy cave. The glowworms of New Zealand and Australia are actually young insects that will grow up and turn into flies. They are the only bioluminescent creatures in the world that live in caves. These glowworms dangle sticky, glowing strands to catch insects to eat. Just imagine. And I'll stop there for now.
Well, thank you, Julia. That was lovely. And I think it whets our appetite for more. And now I have a question for both you and Steve. And that question is, what drew you to bioluminescence? Julia, could you kind of tell us what inspired this book?
Sure. So it was a string of personal experiences that I had, starting with the glowworms in New Zealand. I went there on a family trip, and I was probably in my early 20s and saw the glowworms in the cave, and I was just stunned. But after that, I kayaked in Point Reyes by you, Cindy, and Steve, and saw dinoflagellates in the water, and it was a perfect dark night, and they were very bright. And then was in Taiwan and happened to go on a nighttime walk where I saw something growing on the ground, glowing on the ground, and I had started to become a little bit more curious about bioluminescence by that time, and I realized it was foxfire. And so I think I started to create my own personal art about all of this. I made very simple, like little animation about bioluminescence with a friend, and those illustrations eventually became a gallery show. I can actually show those. And, this doesn't happen often, but an editor saw these paintings online and asked if I wanted to make a book out of them. And so that's how I worked with Greystone and made this book. So it was kind of, as far as the process of making books go, it doesn't really get better than that. A personal project that was picked up and became kind of just exactly a book version of what I had been making on my own. So I was very lucky.
That's very cool. What about you, Steve? What drew you to bioluminescence and, kept you in the field for 30 years?
Well, I grew up in California, so we didn't have fireflies, but they were always somewhat magical to me, of course. And we did have bioluminescence at the beach, so you could go down to the water and have glowing footprints or sparkling footprints and sparkling waves. And I was always fascinated with it, but I went to an engineering school. I didn't really think you could make a career out of studying something like that. But I had a biology professor who was really influential and supportive, and he kind of, I think, saw my curiosity about biology and put me in touch with a colleague of his at University of California, Santa Barbara, who studied bioluminescence. And also there were people there who studied the deep sea and open ocean scuba diving. So I ended up going to grad school in that lab, not really knowing much about the topic, beforehand. But within a month of getting there, I was on a submarine in a cruise off the Bahamas, scuba diving with Peter Herring and Edie Witter and Mike Latz, who are three of the world's experts on bioluminescence. And, it was completely an immersive experience, and I've kind of never turned back from that.
Very cool.
I can show some images of bioluminescence that we've collected. I guess
That would be lovely. Please do.
let me try sharing my screen. So we usually use submarines and scuba diving to collect the animals, because otherwise they get beat up enough that they may lose all their bioluminescence by the time you get to look at them. But we do sometimes use these nets to trawl. And this video is going to show you us emptying out the contents of one of those nets back up on the ship. And this is just a random assortment of things that we collected from the deep. And you can see just how many of them are bioluminescent. You can also maybe pick up the blues and greens that Julia really captured in her book, that they have subtly different shades of bioluminescent emission. And in a study with Severine Martini, we found that 3/4 of the animals that we see from our submarine are bioluminescent, all the way from the surface down to like 4,000 meters, so a few miles down. The right side of this graph is number of things that are bioluminescent. The left side is the number of things that are not bioluminescent. So it's really predominant down there in the ocean. And the way we mostly encounter it is through something like this, like Julia mentioned, the dinoflagellates. This is kind of a dinoflagellate bloom gone wrong, where it was blooming so thick and they all floated up to the surface that it turned the water orange. But each little bioluminescent dinoflagellate is a single-celled organism. And when they make great numbers, you can imagine at night that glowing. And so here's my little volunteer with a bag of dinoflagellates. I don't know if you can see that, but so they're stimulated by physical motion to put off these flashes of light. And we think it serves as a burglar alarm to reveal their potential grazers that are going to eat those dinoflagellates to their own predators. So some of the biggest bioluminescent phenomena are actually made by the smallest organisms. So those dinoflagellates are pretty small, less than a millimeter, but bacteria actually make the display that with my colleague Steve Miller is a satellite researcher. And this blob up here off Indonesia near Australia is a bioluminescent bloom of bacteria that glowed for over a month off the shore of Indonesia. And just to give you an idea of the scale, I mean, for those in the US, but if we transfer this to the coast of California, it would stretch from about San Francisco to Mexico. So it was just an absolutely enormous event made by the smallest known bioluminescent organisms. And we've discovered some animals that are bioluminescent. So some things they're not new species, but nobody knew they were bioluminescent because they just, like I mentioned before, they haven't been collected in great condition. So this is what we call the green bomber worm, which we found has these little green globules next to its head. And fun spinoff of this was that it was picked up in the Octonauts TV show. They depicted this as one of the characters in an episode. So it was pretty gratifying because I think maybe only 2 people in the world have ever seen this glow, in real life with their own eyes. But for them to make this episode, and they actually got the swimming posture really well. Like you would immediately recognize this as one of these green bomber worms. So I was pretty happy that they picked that up, so my son can watch Octonauts and see the green bombers that we found. Another interesting one that we found, this is called a siphonophore. So it's related to jellyfish, but it uses those pulsing clear parts at the top to pull itself through the water. And this one has these lures. They're kind of like a Tootsie Pop. It's got a red coating on the outside, and inside of it's got bioluminescent chemicals. And so these guys eat fish, and they live in the deep sea where there are not that many fish around. So they flick these little lures to attract fish. So it's like an anglerfish, but working in reverse. And another one that we discovered, this was actually on one of those cruises that I mentioned with the submarine with Peter Herring and Edie Witter. I saw a little animal out the window of the sub bioluminesce as it darted away. And it turned out to be the first of these arrow worms. These are really common in the open ocean. They have these hooks on their heads. So if you're a small organism, it would be pretty scary to run into one of these. But from the sub, I was able to see it bioluminescing. And then years, fast forward 25 years, now we have cameras that we can put on the ROV that can film bioluminescence. So I was finally able to capture on video what I was able to see from the sub. And so this is that same little worm. It's about the size of a matchstick, but hopefully you can see. When it darts away, it just shoots out these plumes of light. They make little vortex rings. And this actually is one of the pretty common functions of bioluminescence. When an organism wants to swim away, you can imagine a predator would be completely distracted or be dazzled by that bioluminescent that it leaves in its wake. So you may be surprised to know that not everything you hear on the internet is true. And so here is a web page from Ocean Info, which is a very authoritative sounding place. And they have this picture of a bioluminescent octopus, which is, the first result that shows up if you search for bioluminescent octopus. But of course, that's not the bioluminescent octopus. This is a shallow tropical thing, and those suckers are just white. And even the small factoids about it, the size and everything is not correct. Similarly, if you search for bioluminescent jellyfish, these are the first images that come up, and there's pretty much none of those are real. A lot of them are computer generated. Some of them are non-bioluminescent species. The two with the yellow tilde on them are bioluminescent, but they're kind of enhanced. That's not how the bioluminescence looks. They just made it sort of Photoshoppy. So, this is a minefield for somebody like Julia going out and trying to gather information to make a book. You can imagine, all these illustrations of, here's my book with a page on the bioluminescent octopus that's not bioluminescent, or the bioluminescent jellyfish that's computer generated. And so, one of the things I did create this webpage, this bioluminescence webpage, to try to, put good information out there. But it's really like putting your finger in the dike of misinformation and trying to stop the flood. So I appreciate that Julia really took the time to not just go the internet search route, which I've seen in a lot of other projects, and she really was personally connected to it and reached out to me so that we could have these discussions about what it would look like and what really was going on.
Yeah, it does seem like there's a lot of information and misinformation out there. So how did you find each other?
Do you want to answer? I got an email from Cali, I guess, but I don't know how she specifically found me.
I don't know either. As a complete neophyte, the only contingency for this project was that we work with a researcher. And so when she found you, I had already been looking at your bioluminescence webpage, and there are, your field, it wasn't hard to figure out that you are, you are a leader in your field. And so I was thrilled, but as far as how she picked your name, that's unclear to me. I do have to say, thank you for saying that just now. That was incredibly validating. As an illustrator, I spend so much of my time on Google Images. Yeah. And I looked up that first octopus because it's so beautiful, because I wanted to include it. And so it took me a while to realize that was not bioluminescent. And
it's hard to sift out. Yeah.
Yeah. And this was a really interesting project because, the whole catch is the visual access, right? So I want to make this book because it's hard to physically go to those places. Even as a layperson, the fact that I can count, maybe 4 or 5 bioluminescent organisms that I've seen is incredible. And so those Google images that show ones that are enhanced or not, mislabeled, they were so tricky. And I feel like we should bring up the specimen issue that you and I found. So in order to understand, to draw something accurately, and to draw it without plagiarizing, I want to draw it myself and not, like bite for bite. And to do that, I need to see a couple images of it to understand how it works. So that's the reason that I stayed away from the siphonophore, because like I would need to see a model of it, like turn around and be able to observe it to like actually recreate it. And so as I was looking for multiples of a similar looking creature, I kept coming across bioluminescent organisms that looked pinkish gray. And it wasn't until I talked with Steve that I realized they were all specimen, and that they don't look like that when they're alive.
Like,
they're actually
yeah.
closer to black.
I have some, let me show a slide of that actually. Let's see.
And while you're pulling that up, I think you just mentioned someone named Cali who connected the two of you. Who is that?
Sorry about that. She's the editor behind this book.
Got it. Yeah, I think it's useful for us communicators to know, how these connections get made, how we get the correct scientific information out there.
Mm-hmm.
That is quite a creature.
Yeah, so here's the typical view you would see of an anglerfish. And in Finding Nemo, for example, they have an anglerfish that it has the same thing of these milky white eyes that are completely opaque. And when Julia actually sent me the first drafts of the book, I could see that there was, the anglerfish had these white eyes, because when you look online, that's basically all you find. But really, fish lens proteins are really fragile, and it's called crystallizing. It's sort of like when you hard boil an egg, it goes from clear to opaque. And the same thing happens to the proteins in fish lenses very quickly after, even if it's not been preserved like in formalin or something. So you often see these milky white eyes in deep sea fishes, but if you think about it, it's like not really likely to be functional to be looking through this really occluded eye. But so that was just one of the things that I really appreciated, that she was, she wanted to go the extra effort to make these things as accurate and realistic as possible.
Oh my goodness. It would have been a nightmare to make a book that was factually incorrect. And, I think that's what makes it so daunting for, non-researcher, non-scientist creators, right? Like I don't even need to talk about imposter syndrome, but, the fact that I was able to fact check things and run things by you, it was enormously helpful. You know what? I'm going to grab something to show as part of this real quick.
Okay.
So while I was looking for this book, I was so one of the pages is on the jeweled squid, and I found this stuffy online, and it's like this creator used a specimen as reference, because it should not be this color. It's like the dead pink gray color, which I thought was hilarious. So this is my souvenir from our
They're really
now
beautiful.
it sounds like there's a distinction between specimen and a live organism. Is that correct?
Yeah, when
maybe
she's
I'm
saying
using that
specimen, it's
incorrectly.
like a museum specimen, or something in a jar, not alive.
Not like Julia's dog who just left the screen.
Not a live tail wagger. I just had to take our kittens out. But yeah, I think that was one of the really refreshing things about working with Julia, was just her openness, and we could have these conversations back and forth of like, what's going to be effective artistically and what's going to be accurate scientifically. And I've worked with other books where I reviewed a book for a publisher that was using all photos, and they were just conflating bioluminescence and iridescence and fluorescence, and some of the pictures they showed, they were just like, well, this is just a cool picture, but it didn't have anything to do with bioluminescence. And I finally just said, I tried to talk to them about it, and I just said, don't list me as a consultant and just take my name out of the thing, because they were determined to go forward with it. And that was like polar opposite of my experience working on this with this book.
That is actually a great segue for a question that I have for you, Steven, is, had you worked with an author and illustrator before? Were there any surprises? And I think you kind of talked about the difference between working with Julia on this project and some of your other experiences, but, any other surprises you would care to share?
I've worked with a lot of authors, for magazine articles and for books and children's books and stuff, but I haven't really worked with an illustrator, especially one so accomplished. And I think one of the things, you think that a photograph is like the gold standard of, documenting something, but really there are so many things that we can't see, and that an illustration is way better at, depicting it more accurately than a photograph. So there's so much value in, you can imagine an easy thing of like molecules, like we can't take a picture of DNA replication or something, but you could make an illustration that conveys what you need to know about that. And for bioluminescence as well, even a photo of bioluminescence, like I showed you some videos, but if you take a still photo or a frame of that, it's pretty inscrutable. There's like a little ring of dots, or you kind of don't know where this blob of light is coming from or what the scale is or anything. So having the ability to illustrate and to pour that creative input, I think is so important and kind of empowering, frankly, for our ability to communicate what we can see. So yeah, I mean, it was a better experience than I have had working previously with creators like that.
Fantastic. And now I have a question for Julia. Could you tell us a little bit more about your process? How do you convey these messages?
I thought, thanks for that, Steve. That was maybe the most validating thing an illustrator working with researchers could hear. Maybe I would share 2 images that we worked on together.
Mm-hmm.
One was showing the burglar alarm for the crown of the Atolla jellyfish that, I wanted to make sure that you could see the creature and then the light that they would produce in the same frame to understand how they're related, because I found that to be an inherent problem with, or difficulty in portraying bioluminescence, is that you either see the creature or the light that they produce, but you never see them together, right? Because that's like
Mm-hmm.
counter to how they would be displayed visually. Another one was the jeweled squid and the counter illumination and how they would change based on the light that they're up against. And I was able to work with Steve on how exactly, we tried out a couple of different background variations and how the light would look to make sure the brightness of both the creature and the back looked right. And that was something that I would have never been able to understand on my own, like what is the right luminosity.
Yeah. And I mean, this is a great example of something where if it's working right, that animal is blocking its silhouette by turning on its bioluminescence to match the background. If it's working right, and I've seen this with my own eyes, it just completely disappears into the background. So, it's literally something that you can't see if it's working well, and how to draw something that you can't see. And so we really went back and forth on this to get something that would both read well visually and also be accurate. And on the previous one, I was so impressed by this because we were talking about, well, what should the animal be that kind of triggers this burglar alarm? And I sent Julia some images of different options. And I mean, in my mind, it was like 5 minutes later or something, but it was just like incredibly quickly, she turned back this beautiful drawing of that fish up there. And I was just like, where did this come from? Like, I couldn't believe how quickly and kind of, they're like, when we see them, they're like really cute. And she just really captured the whole feeling of that fish. And it happened like in the blink of an eye as far as I felt. So I was really impressed with the process.
Is an owlfish? Do I remember correctly?
Mm-hmm.
Okay.
Yeah.
Well, it was irresistible. It was the clear choice. We had to show that. But yes, for me,
Yeah.
the challenge is never the drawing, the technical, part. The challenge is the understanding. And so, Cindy, you're asking about process. And I think technically, this book, I kind of explained that the editor found me and because I had already made kind of this like animated story, I had a rough structure in mind, basically like establish what bioluminescence is, dip your toes into the variety and magic of it, maybe pull out for the bigger picture perspective, and then back in real quick for a personal connection. So, you know, the editor and I worked on that text, and then it was a pretty straight shot to the images. And so that's why we've spent so much time on talking about the images, because that was kind of the main like challenge for me. And then just really quickly, I work digitally for all my work. So while I do paint, you kind of saw that in the gallery work, I work all digitally for my books. And for this book, we did consider special inks on top. We did not consider glow in the dark ink because I personally have a bias against them. They're yellowish, and I think that's just not what, that does not represent most of the bioluminescent light that we see. We did experiment with a spot, like a special ink. So usually you have your CMYK and then you get like this special like fluorescent color, but the blue turned out to be pretty similar to this. Oh man, maybe this is too esoteric and more to align with the illustrator side, but we tried a couple of inks, decided that, you know what, good old ink on paper was clear enough. And I almost am very pleased that we came up with this rather simple solution. You know, we didn't need anything fancy. We just needed to show the background and the bright color that would stand against it.
I think your style with the kind of the discrete color palette, it really works well with bioluminescence and also with deep sea, because, you know, I mean, you can see in the image behind me, it's pretty much, you know, it is like a spot color. When the light comes out, it's a very pure color in most cases. So I think it worked really well.
This is so cool. Now I have maybe more of a dramatic question. Were there any tricky spots about working with each other, or, you know, the nature of trying to bridge art and science?
Not for me. I mean, Julia was just so open, and you know, it wasn't like I was telling her what to do, but I feel like you know, I could just look at that and react to the image as an observer, you know, as a fan, and then try to see how to what degree that was compatible with what I knew about the science. And I mean, she was just like, you can kind of tell her personality, but she was super open and free to express her ideas about maybe the challenges of implementing what I was suggesting. But yeah.
Yeah, no, I think because Steve was let me ask all the questions I wanted to, I don't think it could have gone any better, really. You know, I think one thing we've talked about, the more minute, like specific challenges, image to image. But one thing that I feel particularly grateful for is some bigger context. So I think that's really another minefield for someone coming from the outside like me, where you just get all these things out of context. You don't really understand where the field was and where it's moving. And so at the time that I was trying to research, it was either, you know, like the things that I was finding online were either listicles or research papers. And it, you know, I must have missed whatever might exist in between. You mentioned your colleague, Edith Witter, whom you call Edie, and, you know, her book, Below, I think it's called Below the Edge of Darkness, came out after we finished making this book. And, you know, that one book would have provided a lot of context for understanding how hard it was to get these images, like the work that went into simply getting these photographs, and how relatively new this field is. You know, so without that context, it was very like, you're just kind of waiting in the murky darkness. And so for example, one thing that Steve was able to clarify was big picture, like, is the main question for researchers in this field. I thought it was, what are all the species out there that we haven't discovered yet? Because there are, you know, kind of the numbers are mind blowing. There are trillions of these creatures, and there are more that we haven't discovered. And Steve, correct me if I'm remembering incorrectly, but you're like, no, Julia, it's not so much who are these creatures, it's why are they doing it? And that was very illuminating to me.
Yeah. I mean, I think people they do focus on like the new species discoveries, and I showed, some examples of that, but the real challenges left in science, some of it is how do they make the chemicals that produce the light, but really it's like, why is it so widespread? How did bioluminescence get to evolve maybe 90 times across the tree of life? And, things converge, and they have very sophisticated lenses and ways to tune the color and the angular distribution of how the light comes out. And it's so hard to observe the why part of it. Like we speculate, oh, this kind of fish, the males and females have different patterns of light organs, so their photophores are different. So maybe they're using it for mating or for identifying each other. But how do you observe that? How do you actually get a natural, even one data point of bioluminescence being used that way? It's almost impossible. So I think a lot of the residual questions are just, about the whys. And some animals use it for 3 different functions. They may use it to hide, to scare away predator, to attract prey, especially squid. They can have 3 different functions of bioluminescence in one individual. So, it's really hard to track that down. And I think for future researchers, there's we haven't answered all their questions in science. There's plenty of things for people to study and explore.
Yeah, I'm sure that's why so many of the researchers in the audience do what they do, is because there's so much more to explore. So I'm actually curious to ask the audience, and feel free to type in the chat. You can change the "to" in the chat to everyone if you would like to share your answers with everybody. Have you ever worked on a similar project? This kind of bridging of art and science. So while we wait to see what our audience has to say, let's go back to Steve. Do you have any advice for researchers who are interested in working with creatives?
Try to make it happen. I mean, I think, on our cruises, we sometimes are casting about to try to, if we have one spot left on the ship, to try to find an artist that can come and, take a week and join us at sea. Because I mean, I appreciate so much like seeing what people do, these incredible variety of styles. I've worked with a woman who's like really creative painter, but very realistic, but kind of like stylistic. And she actually did a children's book using some of my photos, but it was much less of a back and forth. It was more like, provide some images for her for inspiration. But I just love seeing all the different styles. And I think it's really a way for our science, which is pretty obscure. Our papers may get 20 or 100 like citations in the scientific literature, but then when you get it into the hands of a kid or somebody who maybe is not in the research field, it just amplifies your message. And the other thing is, as scientists, we always we're constraining ourselves by all the caveats of like, you make a generalization and then, but there's all these exceptions. And so you're always like qualifying the statements that you're trying to make about the natural world. And I think having to distill that down in such a way that it can be captured in a work of art is a really good exercise. And it can just amplify the resonance of what you're doing, toiling away in your little darkened lab. To have an artist working with you is really a privilege. So I think I would say do it as much as you can.
And then the flip side of that coin is, Julia, do you have any advice for creatives and communicators who are working with researchers?
I think, beyond what I've shared before about looking for the bigger picture, I almost feel like, just trying to check as much as possible, maintain a good relationship and understand that this is a volunteer, this is something they're volunteering of their own free time. I think when I was talking with Steve, he would just drop little hints of like, oh, you know, we found a, was it woolly mammoth tusk? Like an article about where you found a woolly mammoth tusk on the floor of the ocean outside of the Monterey Bay, you know, just incredible things. And here I'm asking him if this color looks okay. And so I think there's a lot of like, you know, a little, you know, trying not to be afraid to ask, trying to be careful and respectful and polite, but, you know, just understanding the resource that we've been given and how precious that is. I do want to say though, that it has been my personal dream to follow a team of scientists or researchers to one of the poles. And if my artist community knew that, Steve, you sometimes offered up spots on your boats to visiting artists, I don't know how to convey how how special that would be.
We'll have
you
to have a
know?
network of opportunity matchmaking for that.
Yeah, it seems like maybe there's some misconnections out there. But yes, I almost think the challenge is just getting to the connection part. You know, from there, you know, you have, I was lucky to have someone who was willing to work with me and willing to field all my questions, but I just didn't expect it. I just didn't expect someone to be so generous with their time. And I'm not sure why I had that feeling. I don't know if it was past experiences, but yeah, kind of a take what you can get is my answer to that.
I mean, there is a difference between somebody like, you know, I get emails from students, which I'm happy to answer, but sometimes it's like, can you do my homework for me? And, you know, then you're a little bit less inclined to take your own time to do something where they send you a list of questions that maybe their teacher gave to the whole class, and they haven't even tried to search for the really easy ones themselves. And they just, can you send me all the papers about blah, blah, blah. And so, you know, when somebody comes in it with, you know, true, honest intent, then it's totally a different dynamic than just pretty much spamming, you know, researchers with your homework assignment. But yeah, there was none of that with you.
Thank goodness.
And I think that questions are already starting to appear in the chat. So we will now open it up to audience questions, just so that it doesn't get lost in the chat. If you have a question, please use the Q&A feature in Zoom. You can type in the question in the Q&A, and the panelists will all be able to see the questions, and we can go through those. But while we're waiting for some questions to come in, let me check the chat. I think perhaps one of Steve's colleagues just shared something about a polar initiative. So this could be your chance, Julia. And yeah, there's some cool experiences that people are sharing about how they've communicated science. And I see one person has asked, generally, would you speak to the process of illustrating and publishing a science nonfiction book for children or middle grades?
So this is my first nonfiction book that I've written, and I've only written three books, and I've illustrated many others. So I think that this question and an earlier one about Sally Corbin illustrating their book idea, I can give a very, very quick, a quick primer. There are two routes: self-publishing and publishing. Self-publishing gets you the book that you want to make, maybe your vanity book that sounds like a negative word. It's not. The book you want to share with your family and friends, you want to see it done. You want to see it done the way you want to do. It is published out of your own pocket. You use an online like self-publishing resource, of which there are many now, and you will end up with a book in your hands and maybe a couple grand short at the end, maybe a pile of books in your closet too. Traditional publishing, the one that shows up on the bookstores, the one that, you know, kids will access in their libraries, and that is it is harder and it is more rewarding. The gatekeepers are agents and editors, and they are just so just overwhelmed with submissions all the time that it sometimes comes down to pure luck. You need to get your idea in front of one of them in some way or another, and then when they decide that they want to, out of the 20 books they're going to make that year, they're going to choose yours as one of them, then things start rolling. Then we can talk about logistics. I'm actually happy to, I'll put my email here. I'm happy to give anyone guidance. But that is the challenge, creating a manuscript or maybe like a sketch of what you're going to make, a strong one is a very important part that goes into how attractive you look as a competitor going into that, trying to be like picked up. But there's a lot to it, and it's so consuming that it's hard for people to do this on the side. That's why you kind of have people who do this as a career, and then people, like you don't really see like much like many hobbyist bookmakers. But let me put my email here, and anyone who's interested can reach out. I'm happy to talk more individually about share what I do know.
That's very generous. We have a question in the Q&A from Heidi. Can you talk about your publisher's role in this process?
So my publisher was Greystone. I usually work with US publishers. They're called the Big Five. It's like Simon & Schuster, Penguin, Random House, Little Brown. And Greystone is smaller. It's a Canadian imprint, but it is a quality imprint. And so the reason that I love Greystone is that they make very sophisticated picture books. One of my favorite is called War, and that's what it's about. And so, they're interested in the nonfiction area. They're interested in beautiful books. And so that's why they were a perfect fit. So they helped me to make the book that I wanted. If I had signed with one of the other American ones, it probably would be a different book. It would probably be a little bit more narrative. It would look a little bit more like what you're used to. So the fact that I work with them kind of made the art book that I wanted to. So what happened, my interaction with them is the editor will work with me, we'll create a text together, she'll edit my words, and she either art directs me or there is a separate art director who will also kind of oversee the images. So it changes for different creators, but for me, I'm a relatively hands-off worker. Like I basically make sketches, they make one or two changes and they say, okay, go to finals, and I make finals, and there's one, there's not much. But with other people, sometimes it can be a very hands-on process, which can be challenging, but it's one thing to keep in mind as a picture book or a bookmaker is that this is not your product. This is the publisher and your product. So I wouldn't see it as, something that is, there's pitfalls in seeing it as very precious. I hope that answers that question.
I think that there's some interesting comments in the chat. Rebecca shared a social media account called Open Ocean Exploration to broadcast some of these opportunities. So I think another opportunity for Julia, but also people on this chat who are interested in connecting with people in that space. Do we have any other questions coming in?
I have a question for Steve. What's up? What's new to you these days? What have you been excited about in your work?
Our most recent paper was about a month ago, and it's actually about comb jellies and their position in the tree of life. And so there's been this debate for about 15 years now about sponges versus ctenophores. It was always assumed that sponges branched off, then the rest of animals, and then maybe ctenophores, jellyfish, stuff like that. We found that ctenophores had branched off first, and then sponges, and then the rest of animals. And so this some people were not very receptive to that message because it was counter to what they intuitively thought about kind of a progression of complexity. So by sequencing genomes and assembling them to chromosome scale, we got a different line of evidence that also supported ctenophores branching out first. So it's not really bioluminescence in particular, but it did use deep sea ctenophores and sponges as the source of the genetic material to address this very fundamental question in biology. So that's what I've been excited about recently. But we're also working on some bioluminescence project. We have a bioluminescent sea cucumber paper that just came out, and we're working on some protozoans, these little things called radiolarians, single-celled organisms that are in the deep sea that are also bioluminescent. So a lot of little irons in the fire. But
Thank you for sharing.
why you want to make another book?
Oh, yes. that's as we can talk more, but you know, it's very exciting to me. I think that, you know, I think we're on such opposite ends of the spectrum that, you know, you kind of, it's just so shiny, you know, what you do is so shiny and exciting compared to what I do.
I wouldn't say that at all, but
I'm trying
we have
to get,
Oh, no, go ahead. I think there's a lot of mutual admiration amongst everybody here. Yes, it is all very shiny. We have another question that came in, and this is for Julia. If you were starting out your career now, what technology would you want to get to know to support the illustration?
so, you know, I think, I'm guessing that's more of a technical question, like what programs to use. I think that a lot, yeah, I think that, you know, using Photoshop or using, oh, I'm blanking on it, but the program that comes preloaded onto iPads, a lot of illustrators are switching to that because it doesn't require a subscription and can work just like Photoshop and Illustrator. These are things that are just very vital for working commercially because of the turnaround time. I would not be able, I used to paint on wooden boards for my New York Times illustrations, and then I'd stow them under my bed or in my closet, and I had stacks of wooden boards. It's just not feasible to keep doing that. However, I think that it's very dangerous to go straight to digital. Not dangerous. I think that it is very, I would recommend learning to create images in an analog way before going to digital, because digital is it's mimicry. It is mimicking something that is made in real life. And so you lose the context of like, oh, this tool is helping me to create an oil painting like effect without understanding that, like, you know, it is like that because oil paint takes a long time to dry and then you end up smearing on accident. Like it's replicating an accident, but you don't know that because there's no context. And I think just like the foundations are so valuable that I would just make sure that the technical foundations are there. Like, you know how to use a couple different media, you feel comfortable with them, and then you bring them into a digital setting and you work with them that way. Then you will be a formidable creator.
it's funny because there's parallels with science. I mean, I was just thinking for my first papers, I would be like in the darkroom, literally, you know, printing a photo and using rub-on letters and assembling those with like the scale bar with a little rub-on letter and mailing those in a manila envelope to the publisher. And, you know, being able to make even like diagrammatic illustrations and all that has, you know, been completely transformed, sometime in a dangerous way, because now people have been caught photoshopping their results and making fake results. But, you know, between using vector tools for creating diagrams and using photo tools just for making composites, it's like completely changed how we do science as well, or the communication part of science. But yeah, a lot of it starts with a sketch. You know, you start with a doodle or a sketch to figure out like what's going to work in the visualization, and then you implement it in one of these digital tools.
Right. Yeah, and I think the concept behind illustration is what makes us not just makes this job not purely technical. Some parts of illustration are more technical and less, I know there's no judgment value, like it's not positive or negative, it's just, you know, but I think that some really exciting to me parts of illustration are ones where you can be the one who distills the concept, or who problem solves and finds a way to make it more accessible or more easily, you know, And so I think that giving the time to just think about that, look around for where you like it, where you like how it's done is also very valuable.
Oh, hello, Kat.
He wants his opinion to be heard.
Yes. This is the best part of the virtual meetings, not just to connect with people in far-flung places, but also to see their pets. Someone in the chat earlier did ask if Julia illustrates for other authors because they're looking for an illustrator for their book.
I do. I spent most of, that's kind of the bulk of my book work. However, it is harder to get that to happen. There are hurdles. So without elaborating, generally it's hard for someone to ask me and for me to say, yes, I'll be able to do that for you. It's not because I don't want to. It's quite a bit more complicated than that. But I do believe that there will be someone out there who would be the perfect fit.
All right, and I'll give the audience a last call to ask your questions. I know Julia and Steve have both agreed to stay on for a couple minutes longer past the hour, but I do want to be respectful of your time. And while we give people a chance to type in the Q&A or the chat, do you have any last thoughts on, conveying complex scientific concepts in a way that will allow the rest of us to understand, what are some strategies or tips or things to keep in mind?
For this project in particular, I really like Julia's art has such a grace and a quiet elegance to it. And I think it suits bioluminescence really well, but obviously would translate to other topics. But I like the idea that it's just distilling down. It's kind of like stripping away all the distractions and just purely, showing the essential element in a way that's just really there is a really quiet power to it somehow. I don't know how to explain it, but it really worked well for me. So, there are other ways that it could be interpreted, like really, tie-dye, dynamic, flourishing, fireworks. But I thought that this was really suitable for the deep sea and for, even the inside of a cave or foxfire in the forest. He's sitting on a log with glowing mushrooms coming out of it. It's all has just a really nice stillness to it.
Thank you. I think that if I had to add something or answer Cindy's question that way, I guess from the creator side, this is halfway through this project, I realized I should have been looking at videos all along rather than flat images. That was new to me. So, and then I also, I mentioned Edith Witter's book. I don't know how I would have found more resources like that, but had I read it earlier, that would have been very helpful to me. And so it's hard to know what you don't know until it comes up. But just, I guess if I were to speak to my future self, it would just be, I wonder if I would have started my conversation with Steve by saying, I've looked at these resources, what else should I look at? Or I'm relying, just even if I don't know if there's a question in there, I'm relying on images, flat images. Any other recommendations? But again, easier to say in hindsight.
All right, it doesn't look like we have more questions coming in, so we'll get ready to wrap up. If each of you could share with the audience how we can connect more with your work, Steve, how do we hear more about what you're doing? Julia, how do we buy your book or see your upcoming project? If you could share that with all of us.
I'm on Twitter, I guess would be the best way. The bioluminescence webpage is more of a slow update. It's like a reference thing, so I don't post every new paper on there. But probably on Twitter, I guess at BEROE, which is a kind of a comb jelly.
Okay, I'll just put that in the chat. And someone just asked if Julia shared her email. It is in the chat, but you can also find the contact information on Julia's website if you miss it in the chat. And there it is again. So thank you both for your time. Thank you to our audience members for joining us today and sharing your experiences. This has been recorded, so the recording will be available on the lightsources.org website, and everyone who registered will be sent the link later on. So we are so grateful for this time, and we hope you all have a good one.
Thank you so much.
Yeah,
Thank
thanks
you.
everyone
Great to
for
see you guys.
joining.
right.
Thank you.
Bye.